Okay, so now we're talking about toxins and stresses on organisms. And does anybody know offhand what this image is of? You can tell what it is, but or where it was taken.
Is that Mare Island in San Francisco?
No, it's not. Good guess though. So this is the Cuyahoga River in Ohio, and the 1950s, and industries used to just dump their waste into the rivers, and they dumped so much oil, hydrocarbon products onto this river that somehow it caught fire. And here they're trying to put it out. And it didn't happen just once, but this image was really iconic because it was part of the movement towards, you know, controlling what people did with environmental chemicals. And then the other part of that happened was Rachel Carson wrote a book called Silent Spring. And you've probably heard about this by now, but she's sort of a hero of mine and many other people's for many reasons. And her point is that the release of DDT and other pesticides went up through the food chain and killed all the birds. And so you'd have places where there used to be birds and the spring was silent, right? But she did so coming from a scientific background. She was a researcher, and she really thoroughly researched her work and put the science behind. So she's a fantastic scientific communicator who ultimately, I think her work catalyzed the formation of the Environmental Protection Agency in the United States and protection efforts throughout the world. And she also went through a lot of personal issues as she went through all this. So you can read more about that, and you probably will, given what I'm asking you to do this weekend. So this is pretty old, in 2004 National Water Quality Inventory. And I think part of the reason that it's old is that the EPA stopped reporting some of these things by state. And so the data are, you can't quite pull these data out right now. But what it has is a percent of impaired lakes and percent of impaired rivers of all the state reports agglomerated together. And natural impairment is things like floods, droughts, and excretion from wildlife. So there are some that are considered naturally impaired by the regional standards. We see that lakes, mercury is the biggest issue, and that has probably not changed. And PCBs and X, that's probably gone down a little bit. Nutrients have gone up. Metal, BOD, nuisance species probably gone up because there's more of those. And sediment are being the top ones. And then the sources of these atmospheric and agriculture are the biggies. Hydromodification is another one. Urban runoff. So the sources of impairment are on this other axis. The lower left panel has a percent impaired rivers, and we see it flips a little bit where pathogens are a big one. That's coliform bacteria and such, because a lot of our sewage goes into them. Habitat alteration, biochemical oxygen demand. Again, these are both sewage related things. Then nutrients, metals, sediment, mercury. And I think mercury's probably come up since then. And agriculture is the big one. And that's an interesting thing because you see that sewage is important for a lot of them, but not nearly as important as agriculture. Yet, because it's a point source, some of these things are point sources, such as sewage. They're easier to regulate. We'll talk about point sources versus non-point sources, which is just the way the system's set up to regulate pollutants. It makes it more difficult. In this chapter, we're going to go over basic toxicology, bioassessment, acid precipitation, metals, and other inorganic pollutants, nanomaterials, organic pollutants, including plastics, suspended solids, and thermal pollution. So there's a number of basic terminologies that we need to go through. Can anybody tell me what acute versus a chronic exposure means? what the differentiation between acute and chronic?
Acute short-term to chronic is over a period of time.
Exactly. So you get an intense pulse, so that's what happens right away, and chronic is a longer term. So you can imagine that if maybe something's not toxic if you just get exposed to it once, but if you could repeatedly get exposed at that same concentration, it can become toxic. And the responses can be lethal or sublethal, and they can be cumulative. And cumulative then would be the chronic exposure, right? It would lead to cumulative effects. The lethal dose or lethal concentration 50, or effective dose or effective concentration 50 are the ways that people express concentrations or doses to organisms. And that sub 50 is there because, I mean, I guess I should ask you all, what do you think? Why do you think there's a 50? What it means 50% of the population is influence of whatever that population is. Why do you think it's expressed that way? So good. Audra, Abigail's thinking. I can give you a hint. Let's say we have 100 fish and we start slowly jacking up the concentration. Do you think they're all going to die at the same time? Like at one concentration? No, right? Why? Why not? Right, there's varying level of tolerance, right? So organisms vary, right? There's just natural variation, and evolution leads to natural variation. Just like some people are more tolerant to some things, other people are not, right? Some people are more tolerant to allergens and they don't respond at all. Others have an effective concentration that kicks off their allergies terribly. And so that's why this LD50 and EC50 are used, because you want to find out when have some idea when half the population is affected. You may want to use a value that's more restrictive than that, like when 10% of the population, like where you're just starting to see effects, LD10, EC10. And oftentimes with human health, we're way down there, like if 1 in 1,000 people die or something like that from something, then it becomes an issue that we're worried about, or 1 in 100,000. So it kind of depends on the organisms. People care a lot more about people than they do about other things. So if 50% of the diatoms die in a lake, nobody knows it. You're right, nobody cares. If 50% of the people die in a city, then it makes big news, right? Those are kind of the extremes. Like the fish kills, if 50% of the fish die, they might notice it. They're not getting as upset as 50% of the people dying. One of the things that we have these ideas of additive versus multiplicative effects, because organisms are not being exposed to only one toxin at a time. So you can have several different responses. And an additive, you could have a subtractive effect even, where one toxin kind of causes an organism to shut down and then you don't get exposed to the second, so it sort of protects them from it again. Some toxins, if you have half the effect of one and half the effect of other, it just adds right together. And then there's a multiplicative effect, the stress effects, where one toxin stresses an organism, makes them way more susceptible to the other toxin, and vice versa. So you can have multiplicative or synergistic effects. And the effects can also be related to other factors on toxins. So if they're stressed by habitat destruction or something like that, things that wouldn't necessarily be stressful to them, then that might be cause that. There are 3 ways that we can think about movement of toxins into organisms. The first is the bioconcentration. It's just straight moving from the environment into an organism. So for aquatic organism, it would just be the concentration in the water moves into the organism. You can also accumulate from food, so that's bioaccumulation, and that is going to be really more pronounced when in lipid soluble compounds. So if you have a metal dissolved in the water and a fish is passing it across its gills, it can get in quite easily and just get concentrated that way. If you have an organic toxin, then that one isn't really dissolved in the water. It gets stuck in the sediments and all that stuff, and so it's more likely to not bioconcentrate so much as bioaccumulate. And if each food level of the food web concentrates it more, we have biomagnification. That is the increase in concentration. So another iconic species, and the idea of DDT is that it was concentrated as it moved up the food web and weakened the shells of bald eagles and lots of other raptors and harmed other higher predators. But that was iconic because it was going to lead to the extinction of our national bird. And in controlling DDT, that was forestalled, and the numbers of eagles have come up substantially. So even around here, you would maybe never see them, and now we have large groups that you can see in the wintertime along the Kansas River, and some reproductively successful nests that have been established in Tuttle Creek and Tuttle Pond and Milford nearby. That's true in many parts of the country and any other parts of the world where they've successfully controlled these biomagnifying toxins that influence organisms. So here's the bioconcentration factor here, and this probably should be bioaccumulation, but the higher the water solubility of the organic concentration. So something like ethanol is extraordinarily water soluble, and that's why you can have 40% alcohol in water, and they're completely miscible with each other. Others, such as oil, isn't, and like vegetable oil, but if the toxins are not, then they're going to concentrate a lot, because once they get into the organism, they'll continue to accumulate and concentrate. So how do we look at these toxins in the natural environment? And this is a whole field of environmental research and also management relies on bioassessment. And they use native assemblages to indicate the chronic toxicity of things. And actually chronic and acute, that should say. It's useful because long-term effects are difficult to document, and acute episodes may be hard to sample. So you think about what would happen. So here's the fish kill again behind us, right? Once you find out the fish are all dead, it could well be whatever caused the death of those fishes is washed out of the environment. It's not possible to see what the actual concentrations are. But if you come back to that fish community, it's going to be a very depauperate community. So it can tell you when it should be, not depauperate. If it is, then something's been going on for a long time. You can use then the variable susceptibility of different species of organisms to get an idea of what the toxin is, and we'll talk about that a little bit more. Abigail asked, could we look at the data gathered from isotope sampling? Probably. With something like a toxin, you could do sort of post-mortem analysis in that case. That's probably what they do. They look at their guts and they take tissue samples and start analyzing them to see if they can find the toxin. The isotope sampling may or may not help. It works for some things like nitrogen pollution where you can find it. So that's an interesting question. So one of really sort of most basic indices used in streams for water quality is called the EPT, Ephemeroptera, Plecoptera, Trichoptera. And we know what those groups are now because of our earlier taxonomy. And if you look at if there's a high relative abundance of these three groups, they're species that are quite sensitive to either pollutants directly or low oxygen. And so if you have a low amount of Ephemeroptera, Plecoptera, Trichoptera overall, that suggests that the water quality is bad. There's also an index of biotic integrity that's been proposed, and it's assessed by habitats and fish species. This one, again, you go out and sample the fish. So these, when I talked about, there's a lot of research going on with these. That's because each one of these needs to be tuned to the area that you're at. So you can take, for example, we've been talking a lot about Kanza Prairie Biological Station, which is where there's intermittent streams. You expect to have a more depauperate community there because of the extremeness of the physical habitat, not necessarily because it's an unnatural condition. So you might imagine that desert streams have a different index of assessment than, say, a forested stream. And a tropical stream might have a different index than temperate stream, because the organisms that are there and the things that are adapted to are different. So each of these areas has to have their own sort of tuned thing. And then the managers can go out and sample a bunch of rivers, and they say, wait, this one looks really bad. You know, let's follow up and figure out what the problems are here. So it's quite useful. And this is just some relationships between invertebrate diversity and environmental parameters. So in minimum pH, this is the acidic level. And we see that around pH 7, you have the highest number of species found per sample. But when you go into systems that have really low pH, then the diversity falls off. And also those that have really high pH, the diversity falls off. So this could be from acid rain here. This could be from other pollutants, or things like high levels of photosynthesis leading to pushing the pH. Very productive systems can swing back and forth. In this case, this is Shelford's law of tolerance, suggests that there's this sort of middle range of things that most organisms have tended to adapt to. And I don't think it's really a law per se, but it's a generalization. The reason I don't think it's a law is because if you look at the next panel over here, the maximum chloride, we see that most organisms are really not very adapted to chloride. Freshwater organisms are not very well adapted to chloride in their environment. And there really is no fall off at the low end. Maybe if you threw in something like Waldo Lake that we talked about in the last time, ultra oligotrophic, you'd start seeing that. But for the most part, it's such low levels, it's not very effective. In this case, it's a toxin that almost any concentration is going to cause a drop off in diversity. Oxygen is then an intermediate one, where organisms that are really high oxygen or really low oxygen environments, there's very few that are adapted to that. So the diversity is quite low, whereas most are adapted towards sort of the average or saturation concentrations of oxygen. And turbidity is kind of like chloride, where you have no real hump-shaped relationship. So this is just an example of force stressors and how we can use diversity to index the quality of environment. So we'll talk about the sources and geography of acid precipitation, biological effects. Formation of pH starting in 1987, with the redder being lower pHs in the United States, in the continental United States. And we can see when we were in the 80s that the country was really red. But as we're moving forward, it becomes less and less red, because we controlled emissions of hydropower plants. And so this is a problem where we had a real clear environmental problem, and the response was, you should study it more. So we studied it more and figured out something was going on, and the pH increased. And then we can look at this with respect to sulfate concentration. And again, in 1985, we see that sulfate concentration in the industrial regions of the United States being very, very high. And this is related to combustion of coal and fossil fuels. But as the limitations on coal burning plants and industry releasing sulfate into the air were enacted, we can see that the systems got cleaned up, and the deposition is lower. And sulfate's not all the story. Nitrate is also part of the story. And similar thing, although nitrate also is coming from combustion of fossil fuels like gasoline, so they even add nitrogen sometimes. So you can kind of see areas where there's high populations of people, but not offshore winds that stop it. Like this is all coming from LA, right? And this is the Front Range of Colorado. That there's area hotspots of nitrate concentration that are popping up over time. And they did regulate emissions of automobiles as well to some degree, and so that helped deal with that. And the last one's ammonium, which is a little bit different. So ammonium levels are fairly low, and this is not so much related to acid precipitation, but for nitrogen deposition, which we'll talk about in eutrophication. I'm just putting this up now. You can see that a lot of the ammonium deposition is centered on areas that are agricultural and have a lot of feedlots and are fertilizing with anhydrous ammonium a lot. So that's increasing over time. So the amount of ammonium deposition has gone way up over time. And we're seeing that here regionally. Okay. So I mentioned that, you know, this pH was low and one of the, in many of these lakes, and we had a lot of effects. So fish started dying off in lakes in particularly northeastern and southeastern United States, southeastern Canada, and northern Europe. And the claim was it was acid precipitation. And the response of industry and the regulated community was, how do you know these lakes haven't always been acidic? Prove it. So they put some research out to look at that. And these are responses of paleolimnological responses to show acidity. And these Malamonis is a chrysophyte, and it has scales that get preserved in the sediment. And so the y-axis has the depth of sediment here. And you can use isotopic dating to get what the ranges are of those samples. They went to a bunch of lakes and sampled all these different species in the genus, and measured what the pH was. And so you can basically correlate what species are there to what the pH is in present. Then you can go back and see what species were in the lake. So we can see this species seems to be fairly tolerant of low pH, and this somewhat more tolerant, less tolerant of low pH, but still tolerant. This species is not very tolerant at all. So when we have low pH of 6, these species are doing quite well, and the first one is gone, right? We have a pH down to 4.5 to 5, which is where acid precipitation drives it. We have them. So those species dominate. This suggests that acid precipitation is what pushed these systems into a different state, and that historically the lakes were less acidic, and that, you know, sort of put the nail in the coffin as far as regulating industrial emissions, and really led to some effective environmental regulation and slow rebounds of the lakes. This is also harmful to fishes, and the reason for that is the interaction between aluminum and pH. And so high pH, the aluminum is not soluble. Low acidic conditions, many metals, including aluminum, are soluble. However, aluminum is particularly toxic to fishes because it interacts with their gills and interferes with their respiration. And we can see here, there's just surveys of a bunch of lakes, a bunch of streams in northeastern United States, and the aluminum concentrations in the water and in the pH that was there. So as long as you're above 5.5 or 6, then you're okay. And the rain's slightly acidic because when it evaporates and the droplets there, the CO2 goes into the raindrop and through the to the carbonate equilibrium, and it slightly acidifies it. So natural range is about pH 5.5 or a little bit higher. But these are counts of brook trouts, and again, in northeastern United States. And we see at these lower pHs that the number per 0.1 hectares is quite low, but as you get to higher pHs, the numbers come up substantially. In contrast, if you look at the aluminum, when you have low aluminum, you have high brook trout. When you have high aluminum, you have low brook trout. So there's specific data linking the aluminum concentrations to the fish biomass. And then laboratory experiments certainly confirm this, that if you just have a fish in the lab and dump aluminum in, you can, wipe it out. So that's not just, it's not just a correlation.